Hello, everyone, and welcome to today's webinar, Accelerating Discovery Together, Community Resources for Multiplexed Imaging of Diverse Tissues. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more about our sponsor, please visit leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. With that, I'd like to now welcome our speaker, Dr. Andrea J. Radtke, Associate Scientist, Center for Advanced Tissue Imaging, NIAID, NIH. Andrea, you may now begin your presentation.
Well, thank you so much for the very kind introduction. It's an absolute honor and pleasure to be with all of you today, talking about things that I'm very passionate about, both multiplex tissue imaging and sharing this knowledge with all of you and others around the world. And so let's begin. So let's start first with a history lesson and probably also a mispronounced last name. So, here we are in 2023, but hundreds of years ago, Marcello Malpighi recognized the importance of microscopy and medicine. In fact, he was the first to actually apply these techniques to study both human and insect anatomy. So he's credited with applying microscopy to medicine, understandably leading to major advances in human, as I said, and insect anatomy, histology, and plant biology. He is credited with discovering capillaries and the two-compartment system of the human spleen. But haters are going to hate in 2023 and also in the 1600s. And in so doing, he offended his peers. And this is a quote taken from one of the treatments of his work by his peers, saying, "It is our firm opinion that the anatomy of the exceedingly small internal conformation of the viscera, which has been extolled in these very times, is of use to no physician." Boy, were they wrong. And so now in modern day, you can see just the expansion of technologies that now allow dozens of markers to be visualized in human and mouse tissues at single cell and spatial resolution. And that's what I'm going to be talking to you about today. And here's just showing you a human lymph node that was used to create that cathedral of science, that stained glass window on my cover slide, that this is the source data, where you can see the germinal centers in the top right rows and the medullary cords of the lymph node. And so there are many, many different approaches to looking at spatially resolved and single cells and tissues and the architecture of these tissues. And we call these multiplex antibody-based imaging platforms. So today, I'm just going to be focusing on spatial mapping using protein targets and antibodies. And there's many different methods that use fluorophore labeled antibodies. So the little Y in the top left are our antibodies. And so this is a nice summary of the fluorophore labeled methods. There's methods that employ DNA labeled antibodies as summarized in the middle compartment of this figure, and then metal labeled antibodies, and then the methods that are summarized here. And so this just putting in the lower right-hand corner a really great reference that we wrote with our colleagues within the Human Biomolecular Atlas program. And so I just refer you that as a resource for learning more about these methods. And so again, putting back the historical element, in the last 20 years, we've seen an expansion of these technologies. So I've used this timeline to denote the different names of the technologies. And if they're the first description of these in the literature or the founding of a company, and the take-home message is that it's a constant drumbeat, new and new technologies. And so it's very exciting. And 2023, we imagine a year of adoption and expansion through commercialization and also through community efforts to get more and more people using these technologies. And so Marcello and all of you probably realize that these techniques have great promise. And so summarized here that we can look at tissue organization, granular cell types, cell states, stromal and myeloid cells, even discover rare and new cells and look at spatial relationships and cellular components. And I'll show you that throughout the talk. And so, as I said, you can look at anatomical structures just shown here, the purple B cell follicle, the cyan paracortex in the lymph node, We can look at granular cell phenotypes with a different marker expression depending on the antibodies that you use in your panels. We can also look at cell states if you use the appropriate antibody. So we can, here is showing dividing cells expressing Ki67. We can look, because we're looking at intact tissues, we can look at entire meshworks of the folliculodendritic cells, these stromal cells that are undersampled by other means, while also looking at myeloid cells and the relationships with cells, in this case, the germinal center. So you see these beautiful magenta cells gobbling up dead cells in the germinal center. And because, again, we're keeping the tissues intact, we can look at rare cells that you might miss through other methods, and just highlighting that we see these exquisite CD25, these magenta myeloid cells that are at this the T cell and B cell border, and they're making contacts with regulatory cells. So these images can lead to really hypothesis generation. So a lot of promises with these technologies, as summarized on this slide. But nothing is free in life, and so there's a lot of pitfalls in the sense that these technologies require a lot of time. Validating the antibodies can take 6 to 8 months, which I'll show you in the later part of the talk. They're very costly. The average antibody might be $150 to $1,000, and then there's incredible technical challenges and expertise required to really have confidence in the data that is being generated. And so today I've organized the talk to first talk about the promise and applications of this approach, and then two, talk about community initiatives that will really reduce the time, cost, and technical burdens of these methods, allowing more people to participate in spatial mapping. That is really the goal. And so coming back to this slide to just orient you, the methods that I'll be talking to you about today employ fluorophore labeled antibodies, and the method is called Ibex. So it involves cyclic staining and cyclic imaging. And so here's the summary on the left, is showing that by now we've developed both a manual and an automated version of the method. And Ibex stands for Iterative Bleaching Extends Multiplexity. So if you're wondering, that's what it means. And it's a cyclic imaging method where we apply fluorescently labeled antibodies in the top shown there, antibody label, image, dye inactivate, and then repeat. And so you've seen that we validated a lot of reagents, fluorescent probes, diverse microscope configurations, while maintaining all open source and open panel development. And want to acknowledge that science is a team sport. So really pitching the need to work together and acknowledging all the wonderful young investigators, Evelyn, Rochelle, Rebecca, these students who've worked with me over the years, and then my colleagues, Anita, Emily, and Colin, who've been really integral to this, along with members of the Germain Lab. And so highlighting that this is published work now, so you can find this in our first paper, that where we really demonstrated the application of these technologies to looking at mouse tissues and studying immune cells in mouse tissues. Then we've extended it to look at, you can show that Ibex is compatible with animals that have fluorescent proteins. So that's important for some of the work that many of you might be doing, and showing that we didn't see a loss of the fluorescent proteins after rounds of imaging. And then, in fact, you could, with the right multiplexing, you could image around it. In other words, you could keep your reporter proteins as fiducials and then add other antibodies and AF647 and AF Alexifor 700 conjugates to phenotype the cells in greater granularity in those tissues. So a lot of versatility. That's a take-home message. A lot of versatility. And going through this quickly, because this is published, and we refer you to those publications. We've extended this to the human, many different human tissues, showing as a proof of concept anywhere from 16 to 65 plex, and 7 human organs and 6 murine organs, and that's what's published, and certainly a lot more is underway. In the last year, we really wanted to focus on taking the more tedious components of the protocol, which include working from steps 1 to 3, where we're doing the immunolabeling and dye inactivation. In order to scale, we delivered these solutions using a fluidics device. Instead of taking the sample and then soaking off the cover slip before the dye inactivation, we actually keep everything on the microscope stage, and then in summary, what we've created is an open system that has many advantages, both cost and time, while maintaining its versatility and being able to use primary and secondary antibodies and diverse tissue preservation methods, both fixed, frozen, and FFPE. So the overall workflow is, again, we're keeping everything on the microscope slide. So if you look at the microscope and then the blown out version in panel B, we can see that we coat the samples on a coated cover. We section the tissue onto a coated cover slip, have an imaging chamber that you can buy off the shelf. We use a microfluidic system, again, to deliver those antibody cocktails or dye inactivation solution, and then interface control software, which we report in our paper, that allows you to do the antibody labeling, imaging, dye inactivation, all fully automated. And really want to acknowledge the tremendous efforts of Colin Shu, who's a visiting fellow at the time, and James Marr from Leica Microsystems that integrated the microscope with the fluidics system. So again, more details on that are in our Nature protocols. And just showing you the quality of images that we can obtain. So here's an example jejunum showing single cell resolution, both of the cytoplasmic proteins, such as CD49A and vimentin, and the nuclear marker Hurst. And then importantly, and this is a major theme of the day, is that you can capture whole slide regions. So we want to be able to capture very, very large regions, as shown on the image of the human FFPE kidney on the right. And we want to capture these very large regions at high resolution with sufficient dynamic range and in a time efficient manner. And so the typical image properties are for our both manual and automated Ibex methods are shown on the left there. And then because the method is cyclic, so you do these iterative rounds of images. When I look at the figure on the left, showing the stacked images of the red circles, the yellow circles, and the cyan and green, those are to show you that we need to have a common marker, fiducial, which we then align those individual panels to get a full stack of across all the imaging rounds at pixel resolution. And that is we're able to do that through open software developed by Ziv Yanef and Brad Lowenkamp using simple ITK. And we've reported all of this in Zenodo on a public data repository. And so in this first half of the talk, I want to, going through the methods, going through the promise, and now the application. So the first big application was to explore the tumor microenvironment of normal and malignant lymph nodes. This, of course, again, harping on the fact that science is a team sport. You can see there's a lot of authors on this preprint, and I'm really grateful for my NIH colleagues and my Boston Gene colleagues in tackling this challenging problem. And so on the left, showing you that it's well appreciated that there are cell intrinsic and extrinsic factors that govern progression in follicular lymphoma, and really the environment is so critical for the survival and growth of the tumor B cells. And so we used a multi-omic approach to create an atlas of these normal and follicular lymphoma lymph nodes. So on the right, just showing you that in this study, to really advance the technical integration, we're looking at non-follicular lymphoma, we're looking at non-follicular lymph nodes, and then follicular lymphoma lymph nodes, all excisional biopsies, which is, we're grateful to have that kind of quality and amount of tissue to do these assays. We employed multiple different methods, including bulk RNA-seq, single-cell RNA-seq, Ibex, which I just showed you, highly multiplexed and reasonably large regions of interest of 10, on average of 10 millimeters squared. And then I took the most important markers, pared them down to a series of panels that I could apply to much larger regions of interest and FFPE samples. So getting it to be more clinically relevant. And that's what the mix-IF is. And so you're familiar with Ibex now. Again, we're performing the manual method in the imaging chamber using rounds of antibody labeling, imaging, dye inactivation, keeping 1 or 2 markers as a fiducial, showing that little red circle, and then antibody labeling and imaging again. And the markers that we used in our panels are shown here. We've got markers for tumor B cells, 1 for alignment and registration and segmentation, a lot of markers for immune cells, because it's been reported that the immune cells, stromal and myeloid cells, are so critical for tumor progression. And then the asterisk denotes that we applied these markers also to the mix-IF imaging. And so this is just a summary of the individual methods on the left, showing in panel E, the bulk RNA-seq, the single-cell RNA-seq, Ibex, and MixIF. And you can see that we can obtain genomic information from the RNA sequencing approaches. All methods afford varying degrees of looking at the cellular composition of the tumor microenvironment. Clonotypes are really unique for unique information provided by the RNA sequencing, and that the single-cell approaches, both single-cell RNA-seq and Ibex, allowed us granular cellular phenotyping. But finally, to really understand the myeloid, stromal, rare cells, and spatial patterns of these tissues, we needed to use spatial approaches, both the Ibex and then confirmation with the MixIF. And so again, this is published, and I want to give plenty of time for the second half of the talk. So I'll go sort of quickly through this just to show you that when we take the images that are acquired by Ibex and we do object-based segmentation across these 10 samples, and then it's displayed, these individual single cells are displayed in a Hubmap, you can appreciate the very diverse phenotypes and also the richness of the data in terms of the number of segmented cells highlighted in that light blue color with each Hubmap. So you can see across the samples, we can look at flavors of B cells, different tumor types based on the expression of the markers, BCL2 and CD10, IRF4 for signaling, and then many populations of T cells, as I highlighted, the different granular phenotyping that one can do with the right markers, as well as myeloid and stromal cells. And importantly, we can then quantify these across the samples. So showing here that we can look across the samples and, depending on disease progression, for the B cells, the T cells, and then for the myeloid cells, we treated them a little bit differently using a pixel-based tessellation mask to get at the richness of this data, summarizing across the whole samples. Again, these were very much undersampled using other methods, as well as looking at the stromal subtypes I guess it'd be the x-axis, so to speak. We showing you all the different markers that are used for those tessellation masks, and then we're quantifying it across the whole sample. So a lot of information is available to us by applying these highly multiplexed imaging and then using appropriate analytical means to get after insights into tumor progression. And so in the last leg of the tour, really been focusing on extending our initial findings in this cohort to a larger validation cohort. And in order to do this, we partnered up with the CellDive imaging team. And so as I highlighted in this introductory slide, that both are use fluorescently labeled antibodies, and so they share many properties. And so just highlighting as I go further, they're fluorescent-based cyclic imaging. Both CellDive and Ibex use dye inactivation to achieve high multiplex. And so by pairing the two, we were able to use the imager with our dye inactivation solution that is gentle on the tissue and allows us to use those 61 fluorescent probes that we've been testing with many more in the pipeline. Importantly, another point of emphasis, the need to capture whole slide images, but also being open to antibodies from multiple vendors. And so is T-CySIF and CellDive and Ibex all offer you that opportunity, as you'll see why we need to do that. And then we also need to image many dozens of markers in a single tissue section. So I've shown that to you before, and that's why these technologies are attractive. Working with FFPE tissues to be clinically relevant, of course, and then capturing those large regions of interest at high resolution. And so on the right is showing you, these are the CellDive channels that are afforded on the instrument, and then the fluorophores that we can use that we've benchmarked for Ibex. So we can see that this is easy to plug and play, and want to thank the people, Melinda, Michael, Rick, and Prachi, for their help in integrating the CellDive and Ibex. And so here's just giving you a walkthrough. We talk a lot about building the markers and how do we get the images and why do we need, this is probably what many of you are interested in, and why is open the best way to go? So here's a case, the BCL2 shown in blue, which is labeling the mantle zone of the B cell follicles. We are able to, by using both the CellDive and Ibex, use a clone that's preferred by the pathologist. So we have confidence in the data, and then amplifying it to get an excellent signal to noise. So that's very important. But because it's flexible, Ibex, we use direct and indirect antibodies. I could import all that flexibility to the CellDive platform, and we have a beautiful CD8 that's a direct conjugate, and that's compatible with the full panel. Then, showing we're really interested in follicular dendritic cells because that's extremely relevant for follicular lymphoma, showing with this, this antibody, we can use a direct conjugate, we can use a PE conjugate, which is compatible with Ibex, and testing many different antigen retrieval conditions and clones to get the best signal. And so that's why the cost and time comes in, and the expertise is a lot of iteration, a lot of iterating and tinkering, but really pleased with the results in the long run. And then additionally showing the Ki-67, that this particular antibody, we want to look at the cell states. Are these cells dividing? That it was dim. There's a need to amplify. So you can't perhaps achieve this with other methods. And I actually had to use triptavidin, and then that was working with multiple rabbit antibodies. So that's really the anatomy of an image, the nuts and bolts, just with those 4 specific examples. And then in just summary, extending to a little bit more advanced, but still a simple example. So, not to overload you, is just the 10 markers shown on the left. I test maybe 17 different antibodies and ultimately source antibodies from 7 vendors. And it took me a total of 6 weeks. And again, using the pathologist preferred clone, the ability to amplify or use direct conjugates. That's all very important, and that's what's available with those open platforms. In addition to being open, we really want to get after the tumor heterogeneity, which we know is a big component of the disease. And I just show you a very large, zoomed in region of a cyan follicle, so a B cell follicle, pseudocolored in cyan, as I mentioned, and then the beautiful extracellular matrix labeled by Lumican in magenta. And again, with imaging, we can look at acellular components that would be impossible through other approaches. And of course, we can also look at the whole architecture and how they remodel, the stromal remodeling that takes place in many disease states. So another win for imaging. But when I then zoom out and show you that this is just a small region taken from a much larger sample, again, kudos to our clinicians for getting excisional biopsies, you can see that there's great heterogeneity. And so that particular image, there was a lot of extracellular matrix as denoted by the intense Lumican in magenta, but in other regions of the sample, there is no extracellular matrix. So you want to make sure that you're capturing to as best you can the real scenario, and you need to do that through whole slide imaging. And we had in our preprint before integrating with the cell dive, we had shown that in the mix IF, where we could capture much larger regions of interest, how similar was the community composition to the smaller regions that we captured with Ibex. And we did have a pretty good correlation, about 80 to under 95% correlation based on the community composition. So we were happy with that. And so then finally, just showing there's not only just within the tumor credible heterogeneity, there's even more heterogeneity across samples. And so that's what we're working with our validation cohort. And I just visually display to you and pseudocolored and wild colors, just showing you all the different samples arrayed here, that there's differences you'll see immediately appreciate, differences in the size of the follicles, the shape of the follicles, the amount of extracellular matrix, like that these samples are very, very different. And we want to capture that difference and understand why it's different. And then in the hopes of creating better therapeutics or targets for the patients, especially those most vulnerable. So now we could do that again with the cell dive Ibex integration. So now we're transitioning to part 2 of the talk to talk about how, while these are very promising technologies, there's still a lot of barriers. I have 10 plus years of multiplexed imaging experience. That's a big plus, but I really want to share what I've learned from my peers and my colleagues with all of you, so that it can reduce, again, those entries, that you can accelerate discovery by reducing the time and cost of validating antibodies and the technical challenges that come with these advanced microscopy techniques. And so, again, I refer to one of our favorite articles written by our colleagues within the Human Biomolecular Atlas program, that this figure in the perspective nicely outlines, I think, the pain points of multiplex antibody-based imaging. First, want to alert you to the time scale, weeks to months, weeks, months to years, right? That's true. And many of you probably have already experienced that. And so the steps of acquiring those arrays of samples starts with marker selection. You need to have the right markers for your cell types, identify what tissue preservation method you'll be doing, and then find appropriate antibodies. Then step 2, you need to validate those antibodies using appropriate controls. Sometimes you need to amplify and then compare with traditional imaging. So that's all highlighted in step 2. Then you have to work on validating that full panel, showing that there's no nonspecific interactions or interactions between your antibodies, as shown in step 3, and that can take weeks. And then the data analysis, which I don't talk about, and there's many wonderful resources to learn more about that, and that's a rapidly evolving field, even more so than multiplex antibody-based imaging, I think. But I want to acknowledge that that's absolutely true, painful truth, months to years. So that's a field in need of a breakthrough. And so just taking the next few minutes to highlight in the review where you could find resources. So if you're looking to find cell types and what markers you can use to phenotype certain cells in your tissues, there's a lot of great resources. And this is in table S2, where we shared these gene and protein databases for marker discovery efforts. If you're looking for appropriate antibodies for your tissue preservation and method of interest, we refer you to supplementary table S3 that lists the antibody search engines. And so that can be a great resource to you. And then a huge, one of my top questions that I get is, how do you control for autofluorescence? And the answer is you really embrace it. And this is a table S5 that I prepared that shows you all the sources of autofluorescence, which are really painful in fluorescence-based approaches such as Ibex. And, the summary of this figure for me is that ways, influence the biggest impact you can have on autofluorescence is to prevent it by how you preserve your tissue, and I'll show that to you in the next slide. And if you don't have a choice, say you're working with FFPE tissues, then try to do that with computational means or amplify your signal, as I indicated that you might need to do. And then with the cell dive, how they subtract the background, the autofluorescence correction is a huge win. And we do that as well with our Ibex method. So it's a good resource for you guys, this table, I think. And so as I mentioned, if you can control, and so there's a movie on the left, so you can now have control to play this on your end. So if you push the little triangle in the middle, this will just show you a volume image that is acquired with the C3D clearing solution. And the point here is that what I'll show you in the next slide is that how you prepare the tissue is so critical. So over the years, we've developed an optimal tissue preservation protocol within the Germain lab, and showing you that this method involves fixed frozen tissues. It preserves the tissue architecture and cellular morphology, as you've seen that with the images I've shown you. It's a milder fixative, so you don't have as much cross-linking, and you don't need to do antigen retrieval, and you can use many of the antibodies that you employ for flow cytometry, so that also reduces your total reagent cost in the lab, and I think that's an important consideration. We've tested now many more, and you'll see many more commercial antibodies. And we reduce the autofluorescence from common sources, which include red blood cells and tissue lipids. And importantly, these tissues are archivable. And why I wanted to encourage you to play the video is that you can take these tissues that you've frozen, and then you can prepare them using volume imaging later. So it's a good way to archive your samples, and then you could even do more complex advanced microscopy in 3D later. And that's what's in the movie. So now I want to share something that's been under development for 2 years, and we really just released it in the last month. It's the Ibex imaging community, where we really want to share successes and failures. In life, we have a lot of failure, but we don't really have a means to share those failures to teach ourselves and others. And so our guiding principles is shared ownership. And so I'll show you, I'll tell you, and then I'll show you soon. It's failure teaches success. Again, we want to learn from troubleshooting, but we want to learn from others' troubleshooting. Stewardship and democratization is very important. I told you how costly, both in time and resources, these methods are, and we want to share what we know so that we can accelerate discovery for others, while maintaining our commitment to excellence, generating high quality data that everybody has, that we have confidence in the results that come from these images. And then that we can iterate together, that science is a team sport, and when you pair up, you can do a lot more than what you could do by yourself. And so here, just showing you now is our QR code that has a mountain. So if you scan it on your computer, on your cell phone, you should be able to go to the Ibex imaging community website. And the logo is a mountain because the Ibex stands for, that's the method, iterative bleaching extends multiplexity, but it's also a goat that's able to climb exceedingly high mountains. And that's how it feels like with these high-plex imagings many times. So anyway, it's an inspiration to reach the summit together. And so here again, showing you, here's our goat banner. If you go to that Ibex imaging community through that QR code, you'll come upon this static site. But it's not a website. I mean, Ziv, who was instrumental in building all of this, he always tells me that it's not a data set. Sorry, it's not a site, it's a data set. And you can download that data set from Zenodo. And I'll show you how to do that soon. And what is covered in here? Well, so we said we include, we're supporting many different methods. These are 2D methods that what we define as tissue sections that are maybe 5 to 30 micron sections. They could be fixed frozen or FFP or many, many different methods, tissue preservation methods. We do, multiplex 2D. Those are your standard non-iterative, maybe 4 to 8 markers. We also support Ibex, of course, as the name implies, but I really want to use this slide to say it's not just Ibex. We also have 3D methods. So those are the larger volumes, which I showed you in that video that you played earlier, where you can go from 200 microns to 3 millimeters. So there's the clearing enhanced 3D imaging method developed by colleagues in the Germain lab that allow 4 to 8 markers in one image. And then there's the C3D Ibex, which is underway and developed by Hiroshi and Mondo and other colleagues in the Germain lab, where it applies C3D and Ibex to do more markers in the total volume, about 15 markers. So that's very much underway. But this is what you can find in the Ibex imaging community, along with now also the integration of the Ibex cell dive or cell dive Ibex. And so why all these methods is because antibodies validated for one method will likely work in others. And so starting with the multiplex 2D, if you have an antibody for a T cell marker benchmarked in a non-iterative method, it will likely work for Ibex, it will likely work for C3D, and antibodies that work for C3D will of course then work for C3D Ibex or high likelihood. Same thing with Ibex, those antibodies will also work. And then finally the multiplex 2D, many of those target antibodies could work directly for C3D Ibex. However, antibodies that don't work for multiplex 2D, and that's kind of really the stumbling block, will not likely work in others. So that's why we include all these methods, and also just to showcase the work in the spatial biology field. And so, if you have any questions, you can get in contact with us. And so now we're getting to the point where we have a movie. So that's just to remind me that there's a movie on board. So now I'm going to jump to the movie. I'm going to push play here. And yeah, so here's me walking through the actual knowledge base. So as I mentioned, it's a data set that you can have access to through Zenodo, a public data repository. And with the shared ownership, a major point, the guiding principle, if you contribute to the knowledge base, then you are named on the Zenodo entry. And then as I mentioned, it's a data set. We want you to download it. So everything that's statically displayed on that site, you can obtain here, which includes the reagent resources, CSV table, images when we upload more images, the supporting material. Again, with the shared ownership, when you go to the actual site generated from the data set, you can see all the junior and senior investigators from around the world who've already contributed their knowledge to the site, and we're very grateful and appreciative of their efforts. And we also acknowledge that you may not feel comfortable with sharing, especially unpublished work. So only share what you feel comfortable with. And if you have any questions, please reach out. And so here's an overview. We've got our lovely goats, a very inspiring totem, and there's our principles, and really much we want you to take pride in what you are building with us. So you can get those logos and use them in your talks and presentations. Yeah, you see me motioning in the cursor. And so, okay, we'll let it go next. Great. And just the next part, the reagent resources. This is probably where we're going to spend most of your time. These are where you can find reagents relevant for multiplex tissue imaging, again, with the different methods that I described. If you first go and drop down, we support not just primary and secondary antibodies, but many other accessories that are important for multiplex imaging, including blocking kits. We want to add conjugation kits, xenon labeling kits. Here's where you can sort by target. So this is targeting a particular protein. You can see we have many entries in here. Here you can search by target species. I mean, the majority are human because the work of building those, spatial mapping for human reference atlas, but we also have a lot of mouse antibodies, and we want to add, with your help, other experimental model systems. And then the other fields are really pretty much boilerplate antibody metadata, which were created in partnership with our colleagues in the Affinity Reagent Imaging and Validation Work Group of Hubmap. It's important. We make a lot of custom conjugates, so you want to sort for what might be available to you. Support many methods, as I outlined earlier. Tissue preservation, because that's so critical for antibody performance. So we want you to record those details here, and you can search by tissue preservation method to find antibodies that will work for your particular samples. Target tissue, so again, demonstrating that Ibex and many of these methods are compatible with different tissues and tissue states. So many of the antibodies, for example, would only work in a tuberculosis infected sample, and that's provides a good control tissue to benchmark these antibodies. So that's why we want to record that state. The detergent, this is the devil's in the details. We have observed a difference in antibody performance based on the detergent used in your blocking and staining buffers. Antigen retrieval conditions are so critical, as well as for the iterative methods, Ibex and C3D Ibex, the dye inactivation conditions for that fluorophore. We want to provide both reagents that work and then don't work, which is kind of a novelty. And why we say don't, and that's a loaded term, but recommended for those conditions, for that tissue preservation method, for that detergent, for those antigen retrieval conditions. And then we also want to create a mechanism for self-correction where individuals can agree, yes, that worked for me with those settings that you laid out, or no, that did not work for me. When you click on an individual target, you'll see the listing here. This is a supplementary material. You've got our ORCID. We want to give credit where credit is due. So the person who contributed is named there, and a link to their ORCID page. You can have your publications here. So it's a great way to showcase your excellent work. And then, you can also include notes about what did you do to validate this antibody, which tissues, validation, positive and negative controls. You can see the difference here. What I'm reporting is, we love images. So the difference on the antigen retrieval conditions, same antibody, the CD21PE conjugate. If I do pH 6 and 9, I get a different result versus just pH 6. So we want those details here to help other people get the best quality images. And so what was interesting to me is how even in the lab, this is where these antibodies are all validated by the vendor. So we want to appreciate the work that they do in validating that. But if you take the same clone here for CD11B and human tissues, and then you'll watch as I scroll over here in the reagent resources table, that my colleague had validated it using an antigen retrieval of pH 9. And he says that has worked nicely for him. But I tried it with pH 6 shown here and also in the human tonsil. And I don't recommend it with those conditions. So that antibody works when pH 9.5, but not pH 6. And instead, then if I had to use FFPE and pH 6, I would recommend the clones indicated there. So that's the kind of information that's here, along with protocols. And so this just got launched. And so we want to share more protocols, again, of what works, what doesn't work. And then because we're using GitHub to both as a mechanism to share the data, we have a discussion forum. So this is also where you can interact with us through contributing your reagents, but also asking a question. And so that's what's all included in the knowledge-based tour. And in the final minutes, I'll talk about another community initiative taking place in Hubmap. So having talked to you about the Ibex imaging community, that's one community initiative that's really focused on sharing reagents, software, hardware, related to Ibex and other multiplex imaging methods. I want to talk to you about another community effort that is taking place within the Human Biomolecular Atlas program that involves creating these organ mapping antibody panels and addressing pain points shown on the slide, on the part of the slide on the left. And so you can see, as I highlighted earlier, that it requires a lot of expertise and time to identify the right markers to phenotype cells and find appropriate antibodies and then validate those antibodies in the full panel. And so these OMAPs created by the Human Biomolecular Atlas program are collections of antibodies that define cell populations and anatomical structures and human tissues that are relevant for spatial mapping in support of a human reference atlas. And again, these are developed by my colleagues within Hubmap. And so, it starts with the first courageous group to build these 7 OMAPs. That's where we started, and just highlighting the tremendous work of our team on the left with Liz, John, Gloria's team, Elizabeth, Anna, Prescia, and then Ellen, Christopher, and Diane. And Ellen was instrumental in developing all the website and support materials and SOP with me, as well as Diane. And so we're excited that this work will appear in Press and Nature Methods. And so in panel A, just showing we asked our OMAP authors, our experts in multiplex imaging, for your 7 tissues of interest, the intestine, lymph node, lung, kidney, liver, skin, and pancreas, for your various panels, how many anatomical structures and cell types can you phenotype using those panels? And you can see that our multiplex imaging panels may be anywhere from 12 to 52 markers showing the BP. As I highlighted too, it's a lot of expertise to develop these techniques, develop these panels rather, but cost and time. And so we averaged 6 to 8 months to develop an OMAP. And we asked our authors, how much money do you spend on just antibodies alone for your organ mapping antibody panels? And so, it's regardless of the method, whether they use DNA, fluorophore, or metal labeled antibodies, that there's tremendous cost, anywhere from $30,000 to $80,000, due to the rigor of finding and validating antibodies for your method. And however, when we asked our authors, how much does it cost, 100 tests to use a developed OMAP, that cost comes considerably down. That delta is the value of community and sharing knowledge related to spatial mapping. And so using this figure prepared by Diane Saunders to walk through the steps of an OMAP, as this is, we feel, to be a very important resource for the community. As I mentioned before, you first have to find your right markers, and we ask to identify at least 10 protein biomarkers. An OMAP is at least 10 protein targets that allow you to look at the anatomical structures, cell types, and your tissue of interest. There's a lot of great resources for antibody selection, including the Ibex imaging community and the antibody validation reports that are going to be soon to be hosted by the Hubmap. So as well as databases to finding antibodies. Then you have to validate the antibodies, as we mentioned, the rigor that goes into that, and then validate the full panel, which, as highlighted before, involves eliminating those non-specific interactions, looking at the performance of the antibody across cycles, and adjusting exposure times. And so in the end, you have what's needed to publish an OMAP and get it marked with a digital object identifier. It requires an OMAP table, an OMAP description, and the antibody validation reports, along with review. So those are all the details. If you're interested, you can certainly reach out. We'd love to have you. It's open not just to people in academia and government, but also in industry. We have an industry-led OMAP coming soon from Miltenny. And so just highlighting, in December 2021, we had one OMAP. In a few months, we had 7 OMAPs. In the last year, we completely redid the supporting material to get version 2.0, and soon we'll have 6 new OMAPs for a total of 13, with 20 OMAPs coming and vetted for the end of the year. And so with that, I just want to acknowledge the many, many people that contributed to this work, especially Ron for providing such a fruitful environment, and all our support from our colleagues listed here. And I'll take questions, happy to take questions, and thank you very much for your interest.
Great. Well, thank you, Andrea, so much for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. All right, so let's get started. Okay, great. Our first question asks, what are the biggest challenges for adopting Ibex or other methods in the laboratory?
Okay.
Are you with us, Andrea?
Okay. I think I have audio and I think I have video, but honestly, I'm driving blind. Okay, but Kaylee is saying chat. So you know what? Hey, okay, you can hear me. Great. Hello. Okay. Okay. So this will be exciting. It's always a challenge. So thank you guys for tuning in. It's really a pleasure. One of the first questions, it's from one of my friends, so it's a real honor to answer his question. But it's also a failure and success story. So my friend Josh, thank you, Josh, and part of the community asked, "What's the story behind the stained glass window?" Well, I'm really glad you asked it. It's obviously very deeply personal to me. I'll say, because we're in the topic about sharing failures, I created that artwork during a very dark period of my life. It was Thanksgiving 2020. Everybody was pretty depressed, honestly. So just sharing that you can have scientific successes and failures, but also personal successes and failures. And so anyway, that was a way to focus me to work on something quite pretty and impactful. And to me, there's a parallel between imaging and that they both use light to tell a story. The stained glass and imaging both use light to tell a story. And also my friend Stefan, so there's a friendship component there too. He worked and we iterated. He's such a wizard. He put that together in a cathedral. And so that was very inspiring. And then I also want to tell you that it was actually rejected as we submitted as a cover for PNAS for our original Ibex paper, and it was rejected. So, so if for any of you feeling that out there, but then I was pretty faithful, honestly. I was like, you know what, let me just wait. And then it was selected as a cover for Nature Methods and a special issue related to multiplex imaging that we authored with our Hubmap colleagues. So a delay is not a denial. And so it's very encouraging, and I hope you are encouraged by that. So we never hear the story behind the story. So really excellent question. Thank you, Nathan. Is there standard process you follow for selecting imaging channel and imaging round to place a marker in your panel? This is a really excellent question. I wish there were rules, and we kind of want to develop a set of rules. So certainly I can tell you the OMAPs, those organ mapping antibody panels, it's a solved problem. That's how you go through all the iteration. This antibody needs to come here. You need to amplify it, test. So, so that's what we want to achieve, that's the solved proof. But the answer is, many of the things such as with flow cytometry, dim marker, pair it with a bright fluorophore, right? Put it in a 647 or 594 or channels that don't have a lot of autofluorescence. Also become a tissue whisperer. You stare at your tissues for so long, you'll realize, okay, in the lung, for the alexafluorophoridates, you really need to have a banger antibody. You might need to amplify that. And if that's true, you need to put those indirect antibodies in earlier cycles because you don't want to have cross-reactivity with host species. So it's a really excellent question. We do want to, on the imaging community, there's so many, my to-do list overflows and then Ziv's to-do list overflows, but just to have some rules and tips and tricks. So that's a really excellent question. So another one with Nathan with edge effects. So I see your question, Nathan, about asking the technical question with edge effects. I think I'm understanding that to mean that, are you segmenting cells when they're on the perimeter of the image? You know what, I know that we mask the image the area of the image that we actually want to do our analysis on to avoid the edge effect, if I'm understanding your question correctly. But the cool thing about the discussion forum is that I can port all these questions into the GitHub discussion, and we can create a thread there, and hopefully members of the community can help you and offer suggestions. So I appreciate that. And then another question, are we integrating with spatial transcriptomics? Yeah, so I am not personally, but this is, again, the rewards of the community is that there's a very excited and very passionate Italian PhD student, Giuseppe, so shout out, who is, he's reached out and saying that he wants to take this and he wants to push this forward. And so everyone cheer him on and send good vibes and positivity. Definitely, it's very possible. I think there's a consideration about how you fix and prepare your tissue. I know other colleagues in the Germain lab, Vivian and Shushin and Wingjing, are interested in looking at RNA and protein in the same tissues as well. So to be determined, but that's an excellent question because there's really power in integrating, doing multimodal. So And there are other questions. I'll wait for the prompts from the presenter if I should ask those, so I'll wait. Go ahead. Okay, great. We got, go ahead. So the biggest challenges, I think, hopefully, for adopting Ibex, I hope that we spelled it out, that there's some challenges that are just universal to multiplexed imaging. Abandon all hope. No, I'm kidding. But that the time and cost and how we want to work together to reduce those challenges. I think for Ibex, and certainly we could pose this question for members of the community and post-bac students. It's iterative. So iterative equals tedious. I think it's full disclosure. I think the mountain is very apt because it's a climb. With any of these methods, it's a climb, and so that's why. So iterative means you're going to go around and around the mountain, and so I would say that it's simple but not easy. And that's where with these multiplexed imaging, that's where the value of community is. You form a support group. You say, okay, I'm not doing something wrong. This is just how it goes. So my colleague John Hickey in the Nolan lab, Gary Nolan's lab, the Codex fame and Stanford fame, he's done a lot in this space. And just the other day he told me it takes some 20 hours per sample to process an image from start to annotation. So anyway, it's there's a lot of challenges, and we hope to work together to address them. Thank you. Okay, doing great? Okay, again, there's another question. Is community reagent resources to be commonplace in labs with a wide contributor base? I think just to be passionate about it, for one, and also realizing, and this is a conversation that I had with Alexandra Naba, who started the Matrizome project, which is excellent, is that it's you have to love to do it. It's challenging, and these are passion projects or becomes your hobby, because you don't really get funding for these initiatives, even though we can see that they are impactful. So we operate on a skeleton crew, and honestly, my mother has helped so much, and of course members of the community, but my mother, who's retired, a public educator, shout out mom, who's watching, but she likes to say as a hidden figure, she's getting the metadata, the RIDs. So it's just that the challenge, I think, is that we're also busy. We want to work on science that really leads to discovery and get those, papers out. But the time to curate these resources, we don't get more time or money to do it. So you really have to want to do it and then see the value in it. But I'm really grateful for people like you and Josh and others on the call for doing that. So a question from Sarah from Notre Dame. Is the Leica cell dive indispensable in the imaging step? I'm assuming this is the tau-sense function using the pulse laser for single separation. Okay, so first, I'm a government employee, so I can't endorse any technologies. In our papers, we have used the Leica confocal. That was originally the SP8 was used for our Ibex manual. So we were using a confocal system that did not have tau sense. We were using the Leica Thunder, which is a wide field instrument that does not have tau sense. So it's not, the cell dive does not have that functionality, but the more advanced SP8s do. So no, we don't need it. It's not. Instead, we do sequential separation. We choose our fluorophores carefully, and we're working, and we do dye compensation. So to get that high number of markers in a cycle, it is how you set up the image. You can do confocal wide field cell dive, and we want to publish how we set up our image configurations for the confocal and wide field and display that on the knowledge base soon. There's a great postdoc who's working on the dye separation to provide more details on how to do that. So no, it is not. That's a long answer to, and no, the cell dive is not why we use that. And we've used many, many instruments, and we have people using other microscope manufacturers. Okay, so I think we're good. Are there any more questions or? Okay, great. Thank you guys. Really a pleasure to connect with all of you, and check us out in the discussion, the GitHub discussion, and have a lovely day.
Great. Well, thank you so much, Andrea. We did have a few technical issues with our audio here, so she wasn't able to hear me. So thank you so much for going through those questions and getting those answers out. We really appreciate it. So I want to thank you again, Andrea, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Now, before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you had provided at the time of registration. Now, this webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. So we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everyone, take care. Goodbye.